We know that bacteria reproduce by binary fission, and binary fission usually results in an exact copy of the DNA being passed into the daughter cells. So if this is the case, how do bacteria possibly get any genetic recombination? Without any sort of recombination, just binary fission, then all of the bacteria are really going to be clones of each other, and we know that's not the case. Well, the truth is they have various non-sexual means of transferring or recombining information. Recombination is the process of one bacterium donating DNA to another bacterium. The end result is a new strain that's different from both the donor and the original recipient, because some genes get moved over and some genes remain the same. Recombination depends upon extra chromosomal DNA. You'll recall that most bacteria have plasmids of some sort. These genetic exchanges have a tremendous effect on bacterial genetic diversity, as you'll see here soon. It can confer resistance to drugs and metabolic poisons. They can confer new nutritional and metabolic capabilities. And in general, they can increase the adaptation of a bacteria to the environment that it lives in. Any organism that contains and expresses genes that originated in another organism are considered recombinant organisms. Any transfer of DNA that results in an organism's acquiring new genes that did not come directly from the parent organism is called horizontal transfer. That's as compared to the vertical transfer that we would see if the organism is getting genes from its parents. So horizontal, bacteria to bacteria, but not generation to generation. That's vertical. It's this horizontal transfer that allows bacteria to have such great genetic variation. It turns out, though, that we're learning that eukaryotic organisms, including ourselves, also engage in horizontal gene transfer. When we first considered the human genome, we learned that 40 to 50% of the DNA in our genome is not even from human species. So this gene sharing is going on prevalently throughout all organisms, and the transfer is aided and abetted by microbes such as viruses. This horizontal gene transfer usually involves small pieces of DNA, like plasmids or fragments of degrading chromosomes. Plasmids, as you know, can replicate independently of the bacterial chromosome because they have their own origin of replication. Now, once these fragments or plasmids have made their way into another bacterial cell, they then have to integrate themselves into that bacterial chromosome in order to replicate the genes and pass them on to future generations. There are three means that bacteria can accomplish horizontal gene transfer. They are conjugation, transformation, and transduction. Let's take a closer look. We'll begin by looking at conjugation. Conjugation is essentially bacterial sex, where genetic information is transferred directly from donor to recipient. This is the only direct means of gene transfer that we'll be covering. So in general conjugation, we're forming a conjugative or sex pilus. We're dealing with a bacterial cell that has an F factor, and not all of them have an F factor. But here, the F factor is in a bacterial cell. We denote this cell F+. It's F factor positive. A cell that has no F factor is considered F negative or F factor negative. A cell that has the F factor can produce a sex pilus, and this sex pilus will go across and attach to a neighboring bacterial cell. So it's something about the F factor that provides information for growth of this sex pilus. Once it contacts the other cell, the two cells are drawn much more closely together. So again, we have an F positive cell and an F negative cell. The F positive has the F factor, the F negative doesn't. If you have an F factor, then you can form a sex pilus and connect to an F negative cell. Now, in F factor transfer, we might see this go a step further, where the F factor is copied. So now the F plus cell has two copies of it. One copy spools off across the sex pilus. and eventually ends up in the F negative cell, now producing an F positive cell. So now we have two bacterial cells that both contain the F factor. Now at this point, we haven't transferred any of the donor's genes. We've simply just transferred this F factor or fertility factor. However, this F factor could become incorporated into the bacterial genome. If it's integrated into the bacterial genome, then still we have an F factor. So yes, we can form a sex pilus. And then the information from the F factor can spool through into the F minus cell. But now, because the F factor is integrated into the donor cell's genome, a nice portion of the donor cell's genes may also come across into that cell. Those donor genes might cross over and end up incorporated into the bacterial genome. So HFR is high frequency recombination. It's called this because we have a high frequency of genes moving into the cell and recombining with the bacterial's own genome in order to create genetic variation. Let's watch a quick video about bacterial conjugation.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells. A plasmid is a small piece of DNA, separate from the main chromosome, that carries genetic information for such things as antibiotic resistance. The first step in plasmid transfer is contact between the donor and the recipient. The pilus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell. The plasmid then becomes mobilized for transfer when an enzyme cleaves one strand of the plasmid at a specific nucleotide sequence, called the origin of transfer. A single strand of the plasmid, beginning at the origin of transfer, enters the recipient cell. Once inside the recipient cell, a complementary strand to the single DNA strand is synthesized. When donor and recipient cells are mixed together, eventually all of the cells become donors.
And now this one will show us HFR conjugation.
Sometimes the F plasmid becomes integrated into the host cell genome. The host cell is now referred to as HFR, which stands for high frequency of recombination. The HFR is able to transfer some of the host genes to the recipient. The sex pilus contacts the recipient F- cell and pulls the cells together. The donor chromosome is transferred as single-stranded DNA starting at the origin of transfer. Gene 1, which is closest to the origin, is transferred first. Segments of the integrated plasmid are at the beginning and the end of the DNA being transferred. It is theoretically possible for the complete genome and the F plasmid to be transferred to the recipient cell. However, this does not happen because the donor and recipient cells will separate prior to the complete transfer of the donor chromosome. The transferred DNA becomes double-stranded. The donor DNA is integrated into the recipient cell's DNA by homologous recombination. The recipient now carries transferred genes, but remains F-, whereas the donor cell remains HFR.
So naturally there are some important biomedical implications from conjugation. Not only are there F plasmids, but there are R plasmids. R plasmids are more resistance plasmids, or factors. These plasmids bear genes for resisting antibiotics, and they can be shared through conjugation. In fact, they can confer multiple resistances to antibiotics to a particular strain of bacteria. Some other types of R factors also carry resistance to heavy metals like mercury and nickel. And still yet, other R factors are for synthesizing virulence factors, things like toxins, enzymes, and adhesion molecules. All of these will increase the pathogenicity of a particular strain of bacteria. Let's now take a look at transformation. Transformation is capturing DNA from solution. It's an indirect means of transfer of DNA, and it comes about from a really dynamic series of experiments produced by Griffith. He demonstrated that DNA released from a killed cell can be acquired by a live cell. He used 2 strains of Streptococcus pneumoniae. And in these 2 strains, one was a smooth variety that was encapsulated and was virulent, meaning it caused pneumonia. And then there was a strain that lacked a capsule that had a rough appearance and is non-virulent. Here's a summary of his experiment. When Griffith injected the live strain of the smooth bacteria into his mice, the mice died. When he injected the rough strain, which have no capsules, and they're non-virulent, well, naturally the mouse lived. Then he decided to heat kill the encapsulated version. So these have been killed by heat. They're dead bacteria. He injects them into the mouse, and to no surprise, the mouse lives. However, when Griffith combined heat-killed S strain, these are the virulent strain, they've got DNA that's for the virulent strain, but they're dead, he combines them with living rough strain. So these are non-virulent, and these are virulent, but they're dead. Alone, neither of them cause the mouse to die. However, when he injects a mixture of these into both, he finds that the mice die. Not only that, but he recovers live, smooth strain bacteria from that dead mouse. So what's going on here was his question. Well, it turns out that the rough variety have been transformed into smooth variety by free-floating DNA that had been released from these heat-killed S strain bacteria. So transformation is nonspecific acceptance of genetic material by a bacterial cell. Now, it's facilitated by some special binding proteins on the cell wall that help the uptake of that DNA. Some cells are capable of taking up DNA in this way. We call them competent cells. Transformation is a useful technique for certain types of recombinant DNA technology. For example, foreign genes from a completely unrelated organism can be inserted into a plasmid, and then that plasmid can be introduced into the bacterial cell through transformation. I believe this is how we have insulin made by bacterial cells. The pig gene for insulin has been inserted into bacterial cells, and now we can manufacture insulin at an unrestricted rate because the gene has been inserted into bacterial cells. The same technique when we transfer genes into human cells is called transfection, and there's lots and lots of work going on in this area right now, coming up with potential solutions and cures for a variety of different diseases. Now let's take a look at a quick animation of transformation.
DNA transformation involves the transfer of naked DNA into a recipient cell. In the first step, double-stranded donor DNA binds to specific receptors on the surface of a competent cell. One strand of the donor DNA is degraded by nucleases while the other strand enters the cell. The single-stranded donor DNA pairs with an homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. If there are any differences between the nucleotide sequences of the donor and recipient DNAs, the mismatch repair system comes into play. The repair system removes either the donor or the recipient strand and replaces it with the complementary sequence. Since either strand may be repaired, some cells contain the new donor DNA and others have the original DNA sequences. In the laboratory, cells are plated on selective media so that only the transformants will grow.
Our third and final way for horizontal transfer of genetic material in bacteria is through transduction. Although we often think of them as destructive bacterial parasites, bacteriophages, or viruses that infect bacteria, can in fact serve as genetic vectors. A genetic vector is an entity that can bring foreign DNA into a cell. This is the process of transduction. In generalized transduction, we'll see random fragments of disintegrating host DNA accidentally being taken up by phage capsules. Once they burst free from the cell in the process of lysis, these bacteriophages, some of which are now packaged with the host cell's DNA instead of viral DNA, it's an accident, will go around and infect other cells as usual. However, in this case, we're taking the DNA from the donor bacterial cell and injecting it. So some of these phages are defective phages. That is, they're not carrying the DNA that they intended to carry, their own pathogenic DNA. They're carrying another bacterial cell's DNA. Now, this has to happen between the same species of bacteria due to the virus's specificity. One virus will infect a specific type of bacteria. We'll take a quick look at an animation of this.
In generalized transduction, a segment of DNA is carried from one bacterial cell to another by a bacterial virus called a bacteriophage, or phage. The phage attaches to the bacterial cell and injects its nucleic acid into the host cell. A phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage coat proteins are produced. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle carrying the bacterial DNA infects another cell, transferring the bacterial DNA to the new cell. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell then multiplies and carries new genetic material.
Now there is also a process of specialized transduction. This is where a highly specific part of the host genome is regularly incorporated into the viral shell. So it's an accident still, but this results from the phage becoming a prophage. That is, it's inserted itself into the bacterial genome. Once that gene starts to express itself in the lysogenic phase, again, we're inserting the wrong DNA into the viral capsids. So the gene is excised, replicated, packaged into the viral capsids. However, some of those capsids contain DNA that they did not intend to contain, so they are defective. They inject their genome into a recipient cell, and now that cell has some new bacterial DNA. Again, it's an accident on the part of the phage. They're accidentally picking up some of the donor DNA and moving it to a new bacterial cell, allowing genetic recombination. Let's look at an animation of specialized transduction.
Specialized transduction involves the transfer of only a few specific genes from one bacterial cell to another by means of a phage. The lambda phage, which infects E. coli, is a well-studied example of a specialized transducing phage. When lambda phage infects E. coli, the phage DNA enters the cell and then integrates into a specific site on the host chromosome. When an E. coli culture carrying the lambda phage is induced, phage particles are produced. On rare occasions, a piece of bacterial DNA, for example the gal gene, near the specific site of insertion remains attached to the phage DNA and a piece of phage DNA is left behind. The phage that develop are defective because they do not carry the entire phage genome but can still infect other cells. The defective phage can attach to another bacterial cell and the DNA can be injected. Both phage and bacterial DNA now become integrated into the new host chromosome. Only bacterial genes located near the site of integration of the phage DNA can be transduced, hence the term specialized transduction.
Finally, we'll take a quick look at transposons. Transposons are otherwise known as jumping genes. They can jump from one location on a chromosome to another, or from the chromosome to a plasmid, or from a plasmid into a chromosome, or from one cell to another via plasmid transfer. So jumping genes are mobile genetic elements. They move from place to place in the genome through plasmids and viral genomes. They can disrupt genes wherever they land. So if they land right in the middle of a particular coding sequence, that gene is no longer going to be a valid gene. They also could mobilize other genes with them, like some of the genes for antibiotic resistance that we see in plasmids have come from transposons. Transposons contain DNA that codes for the enzymes that they need to move and reintegrate themselves into another site in a genome. Now we'll take a quick look at animation of transposons.
Transposons are segments of DNA that are capable of shifting from one location to another. A transposon enters the cell by being carried on a plasmid. A transposon can then move from the plasmid into the host cell genome. A transposon can move from one site on the host genome to another site on the host genome. When some transposons move, they replicate, leaving a copy in the original position. A transposon can also move from the host genome to a plasmid.
And before we finish, let's have a quick concept check. What form of recombination does the donor cell survive? This is a good question. We know that transduction involves lysogenic cycle, so that means the cell is going to be destroyed in the process. So we can take both generalized and specialized transduction off there. In transformation, the donor cell is dead. That's why its DNA is out in the surrounding environment. So conjugation is the only one where we're going to see the donor cell surviving.